I would like to introduce our next speaker, who is Dr. Garth Ehrlich. Dr. Ehrlich is a professor of microbiology and immunology at Drexel University College of Medicine, where he also leads 2 research centers and the core genomics facility in the Clinical and Translational Research Institute. His research on biofilms and chronic bacterial pathogenesis led to a paradigm shift in the scientific understanding and medical treatment of patients with chronic middle ear infections a few years back. And so he was recruited to study Lyme disease when Borrelia biofilms were identified, as his research is investigating ways to address antibiotic tolerance by Borrelia persister cells. Dr. Garth.
Thanks, Holly, and thanks, Jessica and Tammy, for having me here again. So I have only recently been working in the field of Lyme disease, and so I'm going to start with work that we had done studying other chronic bacterial infections, because I think it's very instructive to see that, in fact, Lyme is not completely unique. There's lots of parallels and lots of things that we can learn from what we've learned in other diseases. So I just want to start with a dedication to Bill Costerton, who is the father of biofilms. He published a paper in Scientific American in 1978, which really started modern biofilm biology, if you will, and actually coined the term. It was called How Bacteria Stick. And Bill actually was very fortunate. He spent his last 4 years as a member of the faculty in my group at Pittsburgh. He passed away in 2012. And the reason I actually work in the field of Lyme is because I was contacted by Julia Wagner, who runs the PA Lyme, which is an affiliate of the ILADS. And she wanted to have Bill come and speak about biofilms to the PA Lyme group. And I had to tell her the bad news that he had just passed away. And so I got drafted instead. So this kind of the subtitle of this talk is Be Skeptical of Received Wisdom. This is Dennis Hopper playing Moses here, because you can get burned. So I always tell my students, don't believe anything I tell you. I said, you got to go out, and find out for yourself and prove it for yourself, because most of what you learn in school is either wrong or incomplete. So when we started out 2 decades, actually almost 3 decades ago now, to study middle ear disease, the theoretical landscape underpinning our understanding of chronic infections was really very limited. And so this is a kind of I'm going to grab the mic here. So I was approached by a young ENT surgeon, Chris Post, who was also a Green Beret, and he walks into my office and goes, "Post." I go, "Okay." He goes, "I want to use PCR, that's a DNA diagnostic technique, to look for bacteria in ear snot." And I said, "Well, I suppose we can, but I'm not sure where I'm interested." And he goes, "Do you have kids?" I go, "Yeah." He goes, "They ever have ear infections?" I go, "Yeah." He goes, "You're interested." So this is me and Chris here. He's an ENT surgeon, and I was actually at the time a virologist. I actually wasn't a microbiologist, upsetting the apple cart here. So this, as I said, this was his question. Why are middle ear infections, in fact, they weren't even thought to be infections, and other chronic bacterial infections such as COPD or the CF lung, refractory to medical management and antibiotic therapy? And so this was a wonderful system actually to work with. Because there's 2 types of middle ear disease. There's acute middle ear disease where kids are crying, they've got, fever and bulging eardrum. And if you tap their ears, nearly 100% of them will grow bacteria, out of that ear fluid. There's a second type, this chronic form, called otitis media with effusion, where they don't have any constitutional symptoms. They're not sick. They just have fluid in their ears, which results like in a 30 to 40 decibel hearing loss, which is a very bad thing because this is little kids are trying to acquire language. And so if you don't get the fluid out of their ears, it affects their language acquisition, which then affects socialization in school and all that kind of stuff. But the dogma had grown up that this chronic form was not infectious, an active infectious process. It was thought to be an inflammatory process, and the reason was because in the vast majority of children who, if you did, again, test the middle ear fluid for these chronic conditions, no bacteria would grow. Okay, so people thought, well, maybe it's an inflammatory response to an infection that was already cleared, but it's not an active infection anymore. So we developed DNA-based assays, PCR-based assays. This is the early 90s now, and this was really pretty new technology at the time. And all we did was we just developed PCR-based assays for the 3 bacteria that were most commonly associated with the acute form of the disease: Haemophilus influenza, Moraxella catarrhalis, and the pneumococcus. And we simply compared our ability to detect them by this DNA method and by culture. And we can just focus on this bottom pie here. And we went to extraordinary lengths to culture. This wasn't like just sending them off to the clinical microbiology lab. We left no stone unturned. And we were actually able to get about a quarter of them positive by culture. But just using this PCR technique for these 3 organisms, we had over 3/4 that were positive by DNA. So this was actually a pretty remarkable finding. And we published this in the Journal of the American Medical Association in 1995, and it was the first of a series of 5 papers we published where we really kind of worked on redefining what goes on in chronic infections. So the next thing that we wanted to do, and it was a valid criticism of our first study, was that, well, DNA is a very stable molecule. And so maybe the DNA is there, but it's not representative of live organisms. So we thought, well, we'll look for its sister molecule, messenger RNA. And bacterial messenger RNAs, if you're not a microbiologist, are extremely short-lived. It's a very labile molecule. Half-lives are measured in seconds to minutes. And so if you could detect messenger RNA, it would be proof, kind of prima facie evidence that the bacteria are alive. And so we had a great medical student who was working with us, Mark Rayner, who went to the OR with Chris and would collect these effusions right into liquid nitrogen so that the RNA wouldn't get degraded. And these are the ones we were interested in. We had 29 that were culture negative for Haemophilus influenza, but were positive for DNA by PCR. So then what we did is we got rid of the DNA. We treated them with DNase, and we looked to see if there was RNA present then. So then we would do a reverse transcriptase step and then do PCR. And 28 of the 29 were mRNA positive and DNA negative. One, we couldn't get rid of all the DNA. But essentially 100% of the ones that were DNA positive were also RNA positive, hence there were metabolically active bacteria there, even though we couldn't culture any of these. They were all culture negative in spite of our best efforts. So I guess this kind of said that. So we've got these conflicting observations with chronic middle ear disease. The cultures are generally sterile. DNA persists. Bacteria recovered from middle ear fluids are metabolically active. Not only did we show that they were producing messenger RNAs, but we later showed that even though we couldn't culture them, that they were producing proteins. There were many types of inflammatory mediators present. And this was the thing which really bedeviled the clinicians, was that they would treat these children with antibiotics. And they could show that the antibiotic concentrations in the middle ear fluid were high enough that all the bacteria should be dead. Okay? And so this was the reason why it was really thought that it was not an infectious process. And of course, you know, the disease does not resolve with antibiotics. And so this was the conundrum. So it was the fall of 1996, and I was reading the front pages of Science, and there was an article about this meeting that had been held in Snowbird, Utah by the American Society of Microbiology. It was the first meeting on biofilms. And there were some comments in there by Bill Costerton, who had been, as I said, had been studying how bacteria grow in hot springs and streams. But there he said one quote, he says, "The resistance to these biofilms to antibiotics is phenomenal." So I'm thinking, maybe what's going on is that bacteria are forming biofilms. I wasn't even sure what they were in the middle ear. And so that began a collaboration with Bill and led to the genesis, you know, of the infectious biofilm paradigm as a pathogenic mechanism to explain chronic bacterial disease. There were other groups at the same time working independently on this. We weren't the only one. There were half a dozen groups all working independently and having no idea that another one was kind of coming to the same conclusion. So it was one of these times like this, the science was kind of right, the time was kind of right for this. So some concepts. Biofilms are the predominant phenotype in all natural and pathogenic ecosystems, it actually turns out. Pure culture planktonic growth is rarely how bacteria exist in nature. And biofilms, as a pathogenic mechanism, serve as a population-level virulence factor. Because one of the things that's really, really important for everybody to understand and to realize is that biofilms are not just a cluster of individual bacterial cells. When the bacteria form a biofilm, it is a more profound metamorphosis than when a caterpillar becomes a moth. Because a caterpillar becoming a moth is a multicellular organism transforming itself into a multicellular organism. When bacteria form a biofilm, you're going from a unicellular or single-celled organism to a multicellular organism. Now all the evolutionary pressure is on a multicellular organism. So you have an organism that sometimes in its life cycle is single-celled, sometimes is multicellular. You ever wonder why bacteria are the only organisms that have never been affected by all the great extinctions? This is probably the reason why, because they are so much more adaptable. They've been around for so much longer. In fact, we think that the original bacteria were biofilms. If you look at the fossils called stromatolites off the coast of Australia, they're about 3.3 billion years old. It's some of the oldest rock in the world. And actually what they are is fossilized bacterial biofilms. And we think the planktonic form actually evolved later as a way to colonize distant sites. Just like you've got a coral reef, the coral in the reef are very protected, they're very safe, and it's only the ones, that go off, as a planktonic form to try to colonize some new site that are, very susceptible, all sorts of predation. So this is a chinchilla, and the chinchilla has been used for quite some time as a model for middle ear disease, and so we adopted this and actually had been using it ourselves for studies before we had the biofilm concept. But we thought, well, okay, so we already got this model. We'll look and see if we put bacteria in the chinchilla's middle ear, does it form a biofilm? And the answer was yes. But at this time we were working with Bill. We didn't have the microscope that he had at the time. He had just gotten one of these newfangled machines, as I said, like 1996 at this time, called a confocal laser scanning microscope. And so this is a picture of the middle ear of a chinchilla. Now this stain is what's called a live dead stage. So everywhere you see is green are live bacteria. If the bacteria were dead, they'd be red. The red that you see are the host cell mucosa, so the chinchilla's cells, okay? And these bacteria have been treated 3 times with antibiotics. Yet you can see that 100% of them are alive. Now we know exactly what strain of bacteria this was, so we know exactly which antibiotic to use to kill it. And yet it didn't kill any of them because they're in this biofilm mode. So what this told us was that what Bill had observed in the natural environments and the streams and stuff like that was exactly the same thing when we're working with biofilms, in an animal. And then a few years later, working with Joe Kirshner at the Medical College of Wisconsin, we showed the same thing with middle ear biopsies from children. So Joe was actually able to go in when he was putting tubes in kids' ears to suction the fluid out. Then he would send us by FedEx the middle ear biopsies, on dry ice. And by that time, we had our confocal microscope. And here you can see these. This was a pneumococcal biofilm. So again, the red are the host cell nuclei. But all the bacteria here, all the little guys, are all green. They're all alive. This child failed 9 courses of antibiotics. There are no dead bacteria here. This is why antibiotics don't work against biofilm bacteria. Okay. So this is what we call biofilms and things resulting from biofilms. We call them population-level virulence factors, because they're not a virulence factor that's associated with single cellularity. It's only present when you have this multicellular form. Okay, you have to have this type of multicellularity. And these population level virulence factors are usually associated with persistence or chronicity, as opposed to like toxins or something like that, which you usually think of something like with anthrax or something like that, something which is acutely pathogenic, like Staph aureus or something like that. And so there's many types of population level virulence factors. There's phenotypic forms, biofilm formation, kind of the mother of them all. There's higher order structures that some bacteria form, like Caserna formation. Some bacteria actually form nanowires for electrical conduction. Quorum sensing is essentially the bacterial cytokine system, so they can communicate among themselves. We've also recently learned that they have other ways of communication very similar to a primitive nervous system. They produce extracellular vesicles, very similar to our synaptogenic vesicles, which transmit neurotransmitters. And so if you hit a bacterial biofilm on the edge with a toxin, an antibiotic for it, it will release these vesicles into the biofilm. They carry information to tell the cells deeper into the biofilm, change your metabolism, bad stuff's coming. So that's why I said, when I tell you there's a multicellular organism, they are multicellular organisms. And there's also genotypic aspects to this population level virulence. And I'm not going to spend too much time talking about this, except to kind of mention the general concept. Most chronic infections are polyclonal. That means there's multiple strains present. And what do we know about bacteria? This is like the other half of what we do in my lab, but I'm not actually going to be talking about today. Is we know that the different strains of a bacteria, even within the same species, have vastly different genomes, called the distributed genome hypothesis. So because they don't undergo meiosis, there's no restriction for the number of genes that a particular strain has. So if you look at an organism like Haemophilus influenza, each strain has about 1,700 genes, okay? But if you look at the species as a whole, there's over 6,000. So there's this distributed genome, or this cloud, okay, of genes that they exchange back and forth. And when you have a polyclonal infection, they can exchange these genes back and forth, creating new strains, so the immune system's constantly chasing a moving target. And we've shown this both cross-sectionally, just looking at lots of strains. We've also showed it following actual infections where we do clonal lineage evolution and see how the strains evolve through horizontal gene transfer in an infected person. So it's not just a theoretical concern. Yeah, so that's what I was just talking about. We call this horizontal gene transfer, where it's basically bacterial sex. And so we've now evolved this concept we call bacterial plurality that embodies the concept that bacteria within a population display heterogeneity at many levels: phenotypic, metabolic, and genotypic. So just to kind of recap this part of the talk, in general, there's no 100% truth, but in general, if you look at the yin and yang of chronic and acute infections, in general, chronic infections are caused by bacteria assuming the biofilm phenotype, whereas acute infections are generally associated with planktonic phenotype, single cell type. Chronic infections are generally polyclonal, that is, there's more than one strain present. Acute infections are generally monoclonal. Often chronic infections are also polymicrobial, that is, you'll have multiple species present. Of course, everybody who studies Lyme disease knows this, versus a single species. And that they generate diversity through horizontal gene transfer among strains and between species. And this concept of generating genetic diversity is very broad for all chronic pathogens. Okay, if you stop and think about it, all chronic pathogens have a way to do this, whether it's HIV or it's trympanosomes. The mechanism, the molecular mechanism that they use for generating diversity is different. HIV has a sloppy DNA polymerase. Trympanosomes have these cassettes of outer membrane proteins they swap in and out. But all these chronic pathogens, doesn't matter if it's a parasite, a bacteria, a virus, they have some mechanism to stay ahead of the immune system. They're constantly changing their genetics. And these dichotomies apply across all medical and dental disciplines. So we started out, studying middle ear disease in the otolaryngology world, and then showed it was the same thing with adenoiditis, sinusitis, tonsillitis. We've worked with the urology groups and gynecology with prostatitis, endometritis, cystitis, vaginitis, orthopedics, osteomyelitis, arthroplastic infections, dentistry, periodontitis, apical periodontitis, endodontitis, whatever you want to call it. So these are very general concepts, okay? So it's not that this is a rare sort of a thing, okay? This is very, very general. But what was interesting to me as a basic scientist in working with the clinicians is each group of clinicians that we would work with, we would have to completely re-educate them again, and it takes like 5 to 10 years to do that. So even though we published all this work in the ENT literature, we started working with the orthopedic surgeons, they looked at us like we had 3 eyes. And what we learned is what you have to do is you have to find somebody, who's a thought leader in that field and co-opt them, And the reason we figured this out is because Chris was a thought leader in the ENT world, and so as soon as we had the biofilm concept, he just bludgeoned the rest of the ENT community to death. I told you he's a Green Beret. And so we learned from that model, and so we've done this, with every field we go into, we find who the thought leaders are, convince them, and let them carry the message. But this explains why it's so hard, to get these concepts, from one field to the other, because our medical system is so siloed, and so that, every Lyme patient you go to, everybody's got these horrible stories, about how it took so long, to get diagnosed. And the reason for that is because, one, this disease, as everybody is a great imitator, so it looks like 1,000 other things, okay? And two, everybody pretty much practices medicine the way they were trained as a resident, And so if you weren't trained as a resident to look for this disease, you're probably not going to look for it. So what are the medical implications? Well, so the medical implications of chronic infections being biofilms are that they're resistant to antibiotics. So basically that means right off the bat, we don't have a way to treat them, okay, medically. And they're also very difficult to detect using culture because they don't like to come out of the biofilm and grow planktonically. Okay, they're happy in the biofilm. So you have to really convince them to come out. And so all the current treatments really are physical. They're surgical. People do debridement. You go to the dentist. They scrape your teeth and gums. If you have an artificial joint, they go and scrape it off. But we don't have medical treatments. For biofilms. There's no FDA-approved medical treatment for biofilms. So this led us to try to get involved in this field, and I just want to take a second here and thank the Bill and Marion Cook Foundation because they are funding this work for us now. Thank you very much. And so what we're trying to do is actually develop the first class of true anti-biofilm drugs. So what I'm going to do is take you through some of the physiology of how we're targeting this. So, with this cartoon here, when bacteria trash talk to antibodies. Oh yeah, you and what course of antibiotics? So what we're trying to do is actually develop a class of drugs that would repotentiate antibiotics against biofilms. Okay, so now what I'm going to do is I'm going to kind of go through why biofilms are resistant to antibiotics. We kind of went through all this, all these clinically important biofilm infections. Obviously, neuroborreliosis may be the most expensive one. We just don't have any known cost because so few people actually get properly diagnosed. But you can see that every one of these things are like billion-dollar problems, multiple billion-dollar problems. Okay. Why isn't this going? Okay. So our target is a protein in the bacteria called REL-A. So this is the REL-A project. It's an update. This is the team that's working on it. This is this wizard graduate student we have, DJ Hall. who is a computational chemist, a synthetic organic chemist, and is now become a biofilm microbiologist. This is Dr. Yaroslav Kroll, who is my lab manager and one of the top biofilm guys in the world. Frank G., who is my chemistry partner, he and I co-mentor DJ, and Noam Cohen, who's an MD, PhD, ENT surgeon, who's our kind of clinical guy on this team. And John, this is a really fascinating case here. John Cahill was an undergraduate working with Frank. And he did some of the original work, and then he disappeared. And I was asking DJ one day last summer, what happened to John? And he goes, you know, nobody knows, but he is paralyzed now, pretty much paralyzed. He can hardly walk. He's lost a huge amount of weight. You know, nobody knows what's wrong with him. He's had all these, you know, neurologic and psychiatric symptoms, and nobody can figure out what's going on. I said, Jesus Christ. I said, go get him tested for Lyme disease. So a week later, he's got a diagnosis from Lyme disease. They put him on IV antibiotics. He's back in the lab. Said our target is this REL-A enzyme. So what is REL-A? So basically, REL-A turns bacteria into stem cells, okay, through production of what's called magic spot. So one of the things you need to understand is we all have stem cells in our bodies, okay? We're multicellular. And what's the major difference between a stem cell and a regular somatic cell in your body? It's designed to live essentially forever. So if you're 90 years old, you've got stem cells that are 90 years old. The difference is they're not trying to divide, they're trying to survive. How do they do that? They massively upregulate their ability to deal with oxidative stress. Okay? Their superoxide dismutase levels go up, their glutathione levels go up, their DNA repair, enzymes go way up. Okay? So when bacteria become persister cells, that's their equivalent of being a stem cell, and that's what they do. They upregulate their ability to deal with oxidative stress. So why would this process of triggering the REL-A enzyme is called activating the stringent response. It is universally activated in biofilms. Why is that? Well, because the REL-A system is designed to change bacterial metabolism as a result of starvation and stress. And so the bacteria that are deep in the biofilm are always starving. They're not getting enough because the guys on the surface, they're getting all the oxygen, they're eating up all the nutrients. So the guys deep in the biofilm, they're going, well, the only way we're going to survive is if we try to stop growing and we just try to survive. The great thing about this REL-A enzyme is it's ancient. There is no bacterium known that doesn't have it. And it's very, very highly conserved across all bacteria. If anybody's into drug development, you want a really highly conserved target to be going after, so it would be broadly effective. As I said, and so it makes them metabolically resistant to antibiotics when you trigger this stringent response. So the idea is if we can inhibit this triggering of the stringent response, then we could repotentiate essentially all antibiotics against biofilm bacteria. Because we know if you take a strain of bacteria and you knock out the gene for REL-A, it will still form a biofilm. But you can treat it with antibiotics and it just dies. That was work that was shown by Pradeep Singh at the University of Washington. So we're trying to develop this new platform technology to hit biofilms. So we have one patent application in. We are working on the submission of a second one now because no one will be interested in licensing this if they don't have intellectual property protection. So we were fairly fortunate in that shortly after we got involved in this, somebody was finally able to get the atomic structure of raleigh. People have been trying for 40 years, and no one had ever been able to crystallize it, and no one still crystallized it because it's a very floppy enzyme, and it's associated with the ribosome. So it's part of a much larger structure. But they were able, with this new technology called cryo-EM, to get about a 3 angstrom resolution image of raleigh, and about a 2.5 angstrom resolution within the binding pocket for nucleotides. And so DJ, our graduate student, essentially taught himself computational biology and developed this pipeline, and he ran 1 million druggable small molecules against this active site. And out of that, we identified 2 classes of compounds that bind in silico more tightly to the raleigh than its natural ligand, guanosine triphosphate. And indeed, then we actually showed in the laboratory that actually was true. And we demonstrated their efficacy in vitro against multiple bacterial species in combinations with antibiotics. So they said these inhibitors are designed to suppress bacterial stress response. This is what you heard me mention to begin with, the compound that's made that triggers the stringent response is this PPG-PP. It's called magic spot because when people first identified it in the 1960s with thin layer chromatography, they had no idea what was going on. Basically, you're taking a guanosine triphosphate, taking the pyrophosphate from ATP and attaching it on the other side of the nucleotide. And so it's a second messenger. If you're familiar with cyclic AMP, it works in the same kind of way. And so this is just some of our first results using repotentiating antibiotics against, using our drug repotentiating antibiotics. I think this was, I can't remember if this was Pseudomonas or E. coli. But what surprised us was that we weren't really expecting a phenotype for the drug by itself, but we actually got one, and it's pretty profound. We're doing this as scanning electron microscopy, and what we show is that it really changes the structure of the biofilm very significantly. You get this much more open structure, okay, to the biofilm than you have with the wild type that has the raleigh. So clearly the stringent response, I said, getting rid of it doesn't prevent them from forming biofilms, but it changes the structure of the biofilm, it makes it much more open. See, there's less matrix. And so one of the problems with biofilms is they're very hard for our white blood cells to get rid of. So we're hoping that because this will lead to better clearance by the white blood cells as well, because the biofilm isn't quite as dense and might not be quite as impenetrable. I was going to skip over this. So after we screened the first 1 million compounds, then DJ got a couple more computational biology programs and actually set up this 3-step computational system. And he also refined the gridding technology within the raleigh enzyme here. So he's now run another 3 million compounds through this pipeline he's got, this in silico pipeline. And from that, we identified another 40 hits. So we purchased all these. They're all available from this company called Enamine, and tested them both in our in vitro magic spot assay and in vivo in bacteria, not in vivo like in a large animal, but in vivo at least in the bacteria. And so this is just kind of the raw data. Here, I'll just point out, so these are the 40 compounds we tested. This is the control here, so we're looking for things that have less or more inhibition. So this is the control. We want things that are lower than that. So some of them you can actually see accentuate, which is not surprising, because you're hitting a target, so you could have an agonistic as well as an antagonistic effect. But the one we were really excited about, obviously, is this one right here, compound 14, because it just about completely shuts down all raleigh activity. And number 2 did a pretty darn good job as well. And this just shows, great, dose response curve here. So we think we've got, probably is actually hitting the binding site. Now, most people don't think of E. coli as a great biofilm model, but as I said, Ehrlich in my lab is a great biofilm biologist, and he studies E. coli C, which is one of 5 classic strains of E. coli that people study in the laboratory. But most people didn't like to study it because it would always stick to the sides of the tubes, and so people would throw it away. So Ehrlich figured, it's actually probably a good biofilm former. So it actually is. So this is looking at its ability to form biofilms versus other major types of E. coli. Cook's K12. So you see here, you get this big, robust biofilm here at the bottom. This is just quantitation. And so we're using this as a model. And the reason we're using it as a model is because to use Borrelia as a model is very hard. It grows very slowly. And it just would take too long to do the experiment. So what we've done, though, is we've cloned the Ehrlich enzyme out of Borrelia, and we're in the process of putting it into E. coli and knocking out the E. coli as one. So that's kind of work in process. And so here we can see, again, this compound 14, which completely shut down the stringent response. It also prevents biofilm formation in E. coli C. There's this tiny little pellet here compared to the wild type. We've got this big pellet here at the bottom. And then this compound 22 reduces, there's still some biofilm. So we're seeing, that a lot of these compounds that affect Borrelia, they do also affect biofilm formation. They make it less robust. So we think this is good because it has its own mechanism in addition to repotentiating antibiotics. And so this is just kind of looking at the amount of aggregation. So this is, the E. coli wild type C, how much huge aggregation you get here. There's no aggregation that you can see when we're using this compound 14. And yeah, so that kind of a summary slide looking at a lot of compounds. So we're also looking at this compound against other organisms in addition to E. coli. We're looking at it against staph and Pseudomonas aeruginosa because we want a compound that's going to work very broadly against a lot of different strains of bacteria. And so this is our C14 compound here versus just media. So there's a statistically significant difference in the amount of biofilm formation. This is not with any antibiotics. It's affecting biofilm formation in Pseudomonas just like it does with an E. coli. But what was really cool about this is this serendipitous effect is that Ehrlich noticed that when he treated the Pseudomonas with this compound 14, they went from their typical kind of green to kind of a yellowish color. Now, Pseudomonas produces this pigment called pyocyanin, and that, in fact, is what gives it this green pigment, and it is one of its major toxins. And so for people who have CF lung, generally they die from Pseudomonas infections. People who have severe burn wounds, they generally die from Pseudomonas infections. What Ehrlich and DJ showed was that at nanomolar levels, the compound 14 is 1,000 times more effective at shutting down pyocyanin production in Pseudomonas than shutting down the Ehrlich response. So we may actually, although it'll probably turn out, we think it's our lead compound as a biofilm potentiator, it may also by itself turn out to be a really good treatment for Pseudomonas. I don't know if I put the slides in here for this. Yeah, so what we did is we just used a simple invertebrate model with Ceterebedus elegans, the flatworm, and we put in Pseudomonas, infected the worms with it, and we're able to show that we could stop the killing of the worms with the Pseudomonas if we gave them compound 14 as well. We got, I guess the movie's not going to run, but basically you can see is like the ones that aren't treated, they're all dead and they don't move. Now we're getting some movement, and the ones that are treated move around a lot, and they're very, very happy. So we were pretty excited by that result, and we didn't have to get any animal rights to have to do that because nobody cares about worms. So as I said, we're cloning the Ehrlich gene from Borrelia and Pseudomonas and staph into E. coli. So we've got to call it cut out, these are the different organisms here, and we're cloning into this vector, which we're going to put into E. coli, so we'll actually be able to kind of test this in proxy, against these different systems in E. coli, which is very, very easy to work with. And what comes next is toxicity testing in cell lines. And then preclinical efficacy testing. We're working with the group at the University of Maryland that has an orthopedic pin model, which is probably one of the most difficult biofilms to get rid of. And so we figured, well, if we can prevent formation of a biofilm on an implanted object, it should be effective against biofilms that are not on implanted objects, because the ones on implanted objects are the most recalcitrant. So we figured we'd try to start out with the most difficult test first. And yeah, so that's obviously, if it passes those tests, then we go into clinical trials. So thank you.